So in the first lecture, we talked about how the innate immune system can prevent infection by just physically blocking or flushing out pathogens. Can potentially kill pathogens using some chemicals like lysozyme or antimicrobial peptides, and can prevent infection by either having other microbes there or allowing those other microbes to kill the pathogens that we don't want. Now we're going to be moving on to complement. Complement is another part of the innate immune system. So everything we've been talking about so far falls under innate immunity. We have barriers, mechanical, chemical, microbiological. Now we're going to add the second part, complement. So what is complement? Complement is part of this non-cellular immunity. If you remember, we talked about dividing up innate immunity and adaptive immunity, and we said both have cellular and non-cellular parts. This is the non-cellular part, or one of the biggest non-cellular parts of innate immunity. These are proteins that are generally floating in our blood and can enter the fluid in our tissues to eliminate pathogens. And there are a number of parts of this soluble non-cellular innate immunity, but we're going to focus on kind of the most common, most well-studied complement. So not to ignore collectins and contractins and all of these wonderful things. They absolutely are very important as well, but we're going to focus on complement as an example of how these systems work. So complement is produced by the liver. This is going to be a common theme in our class, that I believe the whole body is part of the immune system, from those barriers in our skin to our, our mouth and our salivary glands producing all of these things that kill peptides. The liver is actually a huge immune organ as well, and it produces these very important proteins known as complement. The complement, as I said, flows through our blood, leaks out into our tissue, is generally around our body. And complement is a set of proteins that will recognize a problem and do a few different things, but generally kill that problem. Now, complement is complicated. There are so many different proteins, and I will never, ever, ever ask you to learn all of the proteins and all of the steps in complement. There are so many different factors that trigger so many different complexes, and you can just see here, not only do we have C1, 2, 3, 4, 5, 6, 7, 8, and 9, as well as these other factors, you can see that we have C3b and C3a and C3, just way too many. So we are going to generally say we have complement. The one aspect that does come up again and again is C3b, so one part of complement. It's kind of the most important part of complement, and I always remember C3b because I am a huge nerd and had some friends in college who formed a band known as C3b and the Breakdown Products. Yes, we were all nerdy immunology PhD students, but to each their own. Basically, also, everybody in this picture now works in industry and pretty high-powered positions, so I'm sure they would be horrified to know that I generally show their picture every time I talk about complement and C3b. So, complement, nerdy graduate pictures aside, we are just going to focus on sort of what triggers complement to be activated. We won't talk too much about how it's going and what's happening there. C3b, as I said, is going to come up once or twice because that's important kind of for its activation. We'll get there. And then we're just going to talk about how it ends. So don't worry about all of the middle products. We're just going to talk about what triggers complement activation and then what the end result is. So there are three main ways that complement can be activated: the alternative pathway, the classical pathway, and the lectin pathway. And the lectin pathway is also known, I need to get a new pen, as the MLB, or mannan-binding lectin, or mannose-binding lectin, and we'll talk about all of this again. But these are the three main triggers for getting all of these proteins to act together and to actually start doing something. Because these proteins are floating around in our blood, in our fluids all of the time. So what actually triggers them? The alternative pathway, despite its name of being alternative, is actually the main pathway for complement. Complement will float around and will start to basically be activated pretty much anytime it runs into a cell. And so it's kind of a naturally like shoot first, ask questions later. So it says, shows up and it starts to attack. And the only reason that we aren't being killed by complement, killed by our own complement, is that all of our cells have some self ID basically that stops complement from hurting them. So we have a number of proteins on every single cell in our body known as complement control proteins. It doesn't actually say that anywhere on this slide. CCP, complement control proteins. Now, there are a number of different complement control proteins. There are some that are stuck to the cell surface. DAF, DAF, and MCP are two of the big ones. They are always on the cell, and so when C3b, remember that main product of complement, just comes and randomly sticks to the cell and is about to start attacking it, it is inactivated because DAF or MCP basically prevents C3b from being fully activated. There are also other factors, H and I, that are produced in sort of around our cells and tissues that will also inactivate C3b. So C3b is ready to go and cause problems, but our complement control proteins will stop them. The other main way that, we're going to jump to the mannose binding lectin pathway, or MLB, or lectin pathway. And now, mannose itself isn't a PAMP, but mannan, which is a lot of the mannose molecules put together into a more complex carbohydrate, is unique to pathogens, or at least not part of us. So mannose coming together in this very specific pattern on a cell will allow for binding of complement to that cell. Again, we may have mannose around, but it will not be in this specific pattern. So the complement, or this part of complement, this is C1q, you don't have to know that. This part of complement won't bind because we have different patterns of sugars on our cells. Now, when complement binds to mannan, to those mannoses put together in that certain pathway, it activates and starts to get all of the other proteins in and going, and C3b is going to come and do things as well, that will all come together to activate complement fully. So 2 ways we've gotten so far. Alternative pathway will basically start attacking unless our complement control proteins stop it. Mannose binding lectin, another complement protein, will only bind to certain patterns of sugars that are different on pathogens than on our cells. And then for our last one, this is where the lines get blurred. Complement will be activated if antibodies are bound to something. Antibodies we're going to talk about later. Those are part of the adaptive immune system. But if the adaptive immune system has already been activated, there's already antibodies around, complement will still be activated by those antibodies. So clearly, the first time we see something, the first time we are exposed to a new virus, we don't have antibodies to it, complement won't be activated in this way. But eventually, complement stays active throughout the whole process. And once the adaptive immune system does kick in, antibodies stuck to a pathogen will activate complement. So what does complement do when it is activated? Well, it does a few things, but the main thing it does is form a membrane attack complex, a MAC. All of these proteins, starting with C3b, C1q, all of them are going to come together. They're going to activate each other in all sorts of different ways, but they will basically form a ring that essentially pokes a hole in the cell. This is not good. Cells generally do not do well when there are holes poked in them. So just like those antimicrobial peptides disrupted some cell membranes, The MAC is even a more dangerous, like really kind of stabs right through the cell and can kill the pathogen. Now, the MAC isn't the only thing it does. The formation of the MAC, which has led to complement-mediated cytolysis or cytolysis. Lysis is breaking or popping open the cell, one of the main ways. But complement also does mark pathogens for other triggers that we'll talk about later, and signals to the rest of the cells. And next week, we're going to talk a lot about the importance of signaling between cells. Complement or parts of complement can actually, because remember, we're breaking down and having all sorts of things going on, the sort of breakdown pieces are important to warn other people. You can kind of imagine sort of these breakdown products as sort of the gun residue. If you're looking for a problem and you're trying to figure out what happened, like, sure, the bullet is what killed. The bullet was the problem. And so that MAC would be what killed the pathogen. But typically, if there's one bacteria, there's more than one bacteria. And so the evidence of that, residue from the gun or whatever would be those parts of complement, which would also signal to the rest of the immune system that they better be on the lookout. So we've gotten the first two parts of innate immunity now. Our barriers, got the rule of threes here. Barriers with mechanical, chemical, and microbiological. Then we've got complement that is activated three ways: alternative, MLB, and classical pathway. And in fact, I should have added even that complement in the end does three things: kills the cell by that MAC, that membrane attack complex, signals to other cells that there's a problem with those breakdown sort of products. And even if it doesn't get all the way to the membrane attack complex, that activated stuck complement can trigger other immune cells as well.